All right, we are going to cover Chapter 13, the genetic code and transcription. So by the end of this chapter, you should be able to explain and interpret the genetic code, describe key experiments used to deduce it, illustrate the process of prokaryotic transcription in detail, and then contrast that with what happens in eukaryotic cells. So we're going to break this lecture up into 3 different parts. The first part will be the genetic code, the second part will be the basics of transcription, and then the third part will be differences in eukaryotic cells. So we start with central dogma. So this is something you should have all seen before in your intro bio class. But this is the basics of how we think about the transfer of information that is encoded within the DNA. So the DNA, we know, encodes the information, the hereditary material that it can be inherited in future generations. It's the reason that a species is a species, because its DNA encodes the information for creating an organism. But how does that actually get executed? How are the blueprints actually turned into a house? And so the central dogma describes the process. And so you start with a gene, and so DNA can replicate. So this arrow indicates that DNA is able to make copies of itself. So DNA is able to encode whatever is necessary in order to make an exact copy. So we've covered this in the DNA replication chapter about how the complementary binding of the nucleotides allows you to copy the DNA sequence exactly very easily. And so DNA can only make new copies of DNA. And so the information then flows out of the DNA. And so the first thing that needs to happen is that it has to make RNA molecules. And so the process of making RNA out of DNA is referred to as transcription. So multiple RNA types can be produced. So one possibility is the messenger RNA. And so this is a temporary copy of a gene that contains the information to make a polypeptide. A polypeptide is just another name for a protein. But we also know that a gene can be transcribed in order to produce a tRNA, an rRNA, a small RNA, an enzymatic RNA. So transcription refers to the process of making all of those different types of RNAs. But if a messenger RNA is produced, then the next step of information flow is to produce a polypeptide, to produce a protein. So a protein is an enzyme. It might run some sort of metabolic reaction. It could be a signaling molecule like insulin. It could be a scaffolding protein, something necessary for producing a cell wall. There's all sorts of different types of proteins that are produced. Translation is the process of going from messenger RNA into the polypeptide. So we're going to talk more about translation in the next chapter, but for this chapter we're going to be focusing on the process by which a gene becomes a messenger RNA. And we're also going to focus on the process of the genetic code. So the genetic code refers to how the nucleotide sequences in the mRNA encode the amino acid sequences in the polypeptide. So mRNA, messenger RNA, it serves as an intermediate in transferring genetic information from DNA to proteins. Genetic information is stored in DNA. The code that translates it to protein is in RNA. And then the genetic code allows us to take that mRNA sequence and we can predict protein sequence. So this is very powerful for us. So this is part of the reason why DNA sequencing is so powerful, why it's such a useful technique, is because we can take that DNA sequence and we can very straightforwardly predict information about the proteins that get produced by an organism. So if we sequence an organism, if we sequence the humans, if we sequence chimpanzees, if we sequence mouse, we can take that information and we have, with very little effort, we actually know the protein sequences of the genes that they produce. So we can compare the proteins that are produced by mice to the proteins that are produced by humans. Now that we're actually developing techniques to do ancient DNA reconstruction, so we can isolate DNA from Neanderthal bones, and we can use that information to predict the protein sequence of the genes that are encoded by the Neanderthal genome. And so that is very important to help us understand what are steps in human evolution that occurred that allowed us to be different from our ancestors. And the genetic code is the reason why that is possible. So general features of the genetic code, again, I think this should be review. Each word consists of 3 ribonucleotide letters or a codon. So a codon is a set of 3 ribonucleotides in a row. And of course, it's important to remember that the genetic code starts from RNA sequence. So these are ribonucleotides that you're using. It's unambiguous. Each triplet specifies only 1 amino acid. So there are very few exceptions to this rule, but a codon can only indicate 1 amino acid should be incorporated into a protein. It's degenerate, however. So even though each codon specifies a single amino acid, there are multiple codons that signal that encode the same amino acid. So there's multiple ways to encode any given amino acid. And so we refer to that as degenerate. So you should know the definition. You should know what degenerate means. It contains start and stop signals. So part of the genetic code is to indicate this is where the start of the protein is. The stop signal indicates this is the stop of the protein. So an mRNA is actually larger. What this means is that an mRNA is actually larger than the protein that is produced. And so that's because there's parts of the mRNA that are before the start codon, and there are the parts of the mRNA that are after the stop codon. And so those don't actually get incorporated into the protein because of that. It's commaless. So once the translation begins, codons are read with no break. Every 3 codons are read, and if you add an extra nucleotide, you completely change the frame. So there's no indication like these are the 3 amino acids that are part of the codon. It's non-overlapping, so a single ribonucleotide belongs to one and only one triplet. And then it's nearly universal. So if you look at all of the kingdoms of life, and if you look at the genetic code used within those organisms, it's almost completely the same. So there are certain exceptions, but the exceptions are small. There's a few organisms that have exceptions, and in general, most of it is pretty much the same. And so this is one of the indications that there was a single organism a single organism ancestor. So if evolution had evolved life independently multiple times, then we would expect to see completely different genetic codes in different organisms. So if plants evolved completely independently in a brand new evolution of life from mammals, then one indication of that, for example, would be that the genetic code was completely different between them. But we don't see that, so that's good evidence that there was a single ancestral organism that every single thing that lives on Earth now, evolved from. So the genetic code, this handy table, encodes all of the information that we learned about it. And this is the super powerful thing that allows us to take DNA sequence and predict protein sequence for each gene. And so how do we read this? So again, one of the things you should notice is that these are ribonucleotides that are part of the code. So we don't use a T, we use a uracil instead. And so a triplet is 3 letters in a row, and each one can be one of 4 possibilities. So the first letter can be U, A, C, A, G, the second one can be U, C, A, G, the third one can be U, C, A, G. And so all 64 possibilities. So why are there 64 possibilities? Because there's 4 possibilities for each, and since they're independent, 4 times 4 times 4 is 64. So within this genetic code, there's 64 different lines. These are the 64 unique combinations of those 3 letters. And this code just lists what are the amino acids that this triplet would encode. So let's start with one. So let's say we were interested in knowing what CUC was. So how do we use this table? So the first position is on the left, and so we look at this part of the table. The C is the first position, the U is the second. So the top of the table lists the second, and then the C, the third position, is listed on the right-hand side of the table. But in order to use that, you first have to know what the first position is. And so you'll notice I'm going to the C in the second main row of the table, so the sixth row down, but you can divvy this up further based upon the first position. And so this allows me to go to the exact triplet that I'm interested in. So C, and then we're in this square right here, and then C right here. So this CUC is shown right here in the table, and so I look what amino acid is encoded by it, and you'll see leucine is encoded by it. So this particular triplet encodes for a leucine amino acid. So you can look this over. What are some things you should notice? So one of the things you should notice is that it's degenerate. So it's degenerate because there are 6 different triplets that all encode the same amino acid. So this is where the degeneracy comes from. The degeneracy, the level of degeneracy is different for each amino acid. So in the case of leucine, there's 6 different ways to encode it. In the case of something like arginine, right here, there are only 2 different ways to encode arginine. In the case of tryptophan, there's only 1 way to encode tryptophan. So the level of degeneracy can be different depending on each amino acid. You should also notice that there are codons that are dedicated to the stop. So these indicate the end of a polypeptide chain. So there's 3 different codons that can be used for that, a UAA, a UAG, or a UGA, and those all indicate to stop incorporating amino acids and release the polypeptide chain to become a protein. There's also an amino acid used as the initiator codon, AUG. However, you should remember that this one is a little bit funny. So this one can be used, this is an example of when we go back to the unambiguousness, this is a little bit of a violation of that. So it's a violation because this AUG can be used either as the initiator codon, so that's what the purple means right here, or it can be used, and it's the only codon that can be used to encode methionine. So methionine is another amino acid. So just remember, AUG can either indicate the start, or it can indicate just a normal methionine to be incorporated in the polypeptide chain. And what determines that is contextual information upstream, typically upstream of the AUG. So if there are additional bases upstream, then that can be used to indicate this is the initiator site. If there are not that contextual information, then it's just used as a normal methionine site. So just remember that AUG is special in that way. And that is not true for the stop codon. So if the stop codon is there, it can only be used as a stop codon, termination codon. All right, so there's little tolerance for error. So here we have a set open reading frame right here. And if we break this up by triplet, you'll notice that these are the breaking points right here. Okay. And this right here, if we look at what this codon encodes, it's a GAG. And so we go to our table, G, A, and then down here is G, and so this would encode a glue. And so each one of these codons would all encode a glutamine. So there'd be 5 incorporated here. So we said that it's non-overlapping. So what do we mean by non-overlapping? We mean that this G is only used as a codon once, and this A is only used as a codon once. So GAG, so we have a glutamine, and then we go to the next 3. So if it wasn't that way, one possibility is that GAG encodes the first amino acid, and then AGG encodes the second amino acid. So you can imagine reading it 1 base at a time. But this is not how it is. You read 3 bases, and then you shift over to the next 3 bases. And so what happens if you insert a nucleotide? So here we have now added a nucleotide, a C nucleotide, into the DNA sequence. And so now what happens to the DNA in this case? Sorry, what happens to the protein that gets produced in this case? So the frame is shifted because we have GAG, but now we have GAC, and because we've inserted this, the GAGs that used to be in frame now are out of frame. So now we have GGA, GGA, and GGA. So the remaining ones are out of frame. And so this is what we mean when we refer to it as commaless. So one possibility was that there was something that would help us make sure that we know it's the GAG that we mean to be the codons that are used. And so you can imagine something like GAG, and then you have a comma. So let's say AAA was the comma. And so the 3 A's must be read in order to say, okay, this is the first codon, and now this is the comma, and so now we're going to read the second one as well. But this is not how it happens, and so if you have a shift in the frame, then the problem is that it translates into a completely different amino acid change. And this is not just where the insertion occurs, but everything downstream as well. So all of those amino acids downstream of it are going to be changed by that frame shift. And so this is just what I mean where there's little tolerance for error. So when you look at DNA sequence, if you ever get your DNA sequenced and they searched your exons and they found that there was a 1 base pair insertion or a 1 base pair deletion, they would probably flag that as potentially a very lethal, or not lethal, but a very deleterious type of change that potentially could cause disease. So that's one of the types of genetic changes that human geneticists now look for if they're trying to predict the genetic basis of a rare disease, because they know any sort of insertion or deletion will have a huge effect on the majority of the protein. So now let's talk a little bit about cracking the genetic code. So what were the experiments that allowed researchers to actually create this table right here? So one of the first things that they had to do is figure out how many nucleotides in a row do they need to worry about for this code. So one possibility is that, instead of there being 3, there could have been 4. So maybe 4 nucleotides specified an amino acid. And so the reason that they settled on 3 was just some theoretical considerations. And so they knew that there were 4 nucleotides. They knew that there were 20 amino acids. And so they asked how many nucleotides, what's the minimum number of nucleotides that would be necessary in order to encode 20 unique amino acids. And so let's say we look at 1, and so instead of the genetic code being a triplet, it was just a singlet. And so, you could have a genetic code where A, C, G, and T each encoded different amino acids. So maybe A encoded lysine, C encoded isoleucine, maybe G encoded leucine, and maybe T encoded alanine. But of course, if this was the case, then we wouldn't have any way to specify the other 16 amino acids. And so, this consideration just led for the researchers to say, okay, it cannot be a singlet. It can't be 2, because if we calculate how many possible combinations there are of 2 nucleotides, there's only 16, and 16 is less than 20. 3, however, we can now do 4 times 4 times 4, so there's 64 possibilities. And so the researchers honed in initially on this idea of a triplet code because that was the minimum number of nucleotides that would be necessary. It's also possible that 4 were used or 5 were used, and the code was just a lot more degenerate than it is currently. We know now that the triplet is actually true, but the reason that we can exclude 1 nucleotide or 2 nucleotides is because they couldn't encode enough amino acids. So then what did they do next? So what were some of the experiments that they used in order to try to crack the code? And so, like all of genetics, you should get used to the idea that researchers have isolated some sort of useful enzyme that does something that they can then take over in order to perform some sort of cool experiment. And so in this case, they have an enzyme called polynucleotide phosphorylase. And so what this enzyme can do is it can join nucleotides together. And so if you have ribonucleotide diphosphates, and then you add polynucleotide phosphorylase, you are able to join these together into mRNA strands. So again, these are ribonucleotides, and then you can join these together into strands, and then you release inorganic phosphates. So you notice, what you're using is not the triphosphates that is normally used. These are diphosphates. So this is an enzyme that is specially made in order to join these types of ribonucleoside diphosphates together. And so how is that useful? So you can start out with just using homopolymers. So RNA homopolymers are RNA nucleotides with only one type of ribonucleotide. And so they could create this, so they could chemically synthesize one of these ribonucleotides. So instead of being RNDP, you would synthesize RADP. And so now if you take this and then you add the polynucleotide phosphorylase, you create a homopolymer where every single nucleotide is the same. So this is the first step of cracking the genetic code. So what happens if you did this with all U's? So the artificial mRNA that you created was all U's, and then they measured the incorporation of a specific amino acid, which was phenylalanine. And so this is the first step. This is the first square of 64 that you can now enter. So U, let's find this on the genetic table right here. And so now we know it's a phenylalanine. So you could do this for all 4 different nucleotides, U, A, C, and G. So the next step is they created RNA heteropolymers. And so again, this was the same idea, but instead of adding a single ribonucleoside diphosphate, they added 2 different ones together. So 2 different ribonucleosides were added together. And further, they added them in different proportions. So they didn't add them as so one possibility is, so 1 part RADP and 1 part RCDP. And so that would be 50-50 A's and 50-50 C's, 50 A's, 50 C's. But they also, did experiments like this, where they might add 1 part ADP and 4 parts CDP. So now we know there's a lot more C's in this tube in order to form these single strands of mRNA. And sorry, I skipped a little bit of a step. And so one of the things they also were able to do is they were also able to have an in vitro ribosomal system where an mRNA was able to actually produce a polypeptide. So this is an example here of what would happen when you added a distinct amount of A's and C's. And so in this case, you did 1 part A to 5 parts C. So for every A nucleotide, there was 5 C nucleotides. And so now you can lay out all of the different triplets that can be formed between just using the A's and C's. So one possible triplet is AAA, CCC is another homopolymer, and then you have 3 different combinations of 2 parts A and 1 part C. So you can either have AAC, ACA, or CAA. But all 3 of these situations, you have 1 C to 2A. And then in this case, you have 2 C's to 1A. And so the possible triplets for that are ACC, CAC, and CCA. So you can use simple probability rules to predict how often each of those triplets would form. And so because the A's, there's 1/6 of the mix is A's, then if you want to calculate the probability of 3 A's in a row, it would just be 1/6 to the third power. And so that's about 0.4%. For these three, the possibility of that is 1 to the 6 squared times 5/6. For this one, it's 1 to the 6 times 5/6 squared. And then for this last one, it's 5/6 to the third power. So the probability of CCC is this. And so now they run this reaction. They have these really complicated long RNAs. There's a large number of these. They have the ribosomes go, and then they see which sort of amino acids are incorporated into the protein. So one of the first things you notice is that now we've gone from 1 amino acid to 6 different amino acids that are getting incorporated into the protein. So lysine, glutamine, asparagine, threonine, histidine, and proline. And they also can measure the amount of each of those amino acids that gets incorporated. So lysine was less than 1%, proline was more than 69%. So we already knew from the homopolymers that AAA produces lysine, and we expect to see a very small amount of that, 0.4%, and that looks good. We also knew from the homopolymer work that CCC produces proline, and so we would expect there to be at least 57.9% proline. And so proline, there was actually 69%. And so how do you interpret that? Well, you interpret that as we know CCC is one of the codons that can produce a proline, but there also must be another one as well. And so if you add up the 57.9% and you ask, if there's one of these other codons that might produce a proline, what makes the most sense in terms of the amount being generated. And so we know that for this combination, for each of these, there's about 11.6%. We expect to see 11.6%. So now if we just add these together, 57.6 plus 57.9 plus 11.6, and add them together, then we get something like 69.5% is how much we expect to see. And so this is the interpretation of what makes proline, is that there must be one CCC codon, and then there must be a second codon that is also able to produce it, and it has 2 C's and 1 A's. So let's go back to the genetic code and see if this is true. So we know proline, so we go to the part of the table right here. So we already knew CCC produced proline, and so now we also know that there must be a codon CCA, that a 2 C's and 1 A, and so we see indeed there is. So CCA also is able to produce proline. So we notice that there's 2 other ones that also can produce it. However, we do not expect to see them in this case because we're only combining the C's and A's, and the other codons would require a G or a U. So this helped kind of refine it, but you'll notice that you have no way of knowing, which codon is which. So glutamine, we know if we match up the amount that's produced, we expect there to be one codon that is 1 part C and 2 parts A, but we don't know which one of these is the glutamine. We don't know which one of these is the asparagine. And so these experiments were able to further narrow down what was going on and further provide data of how the genetic code operated, but they really needed to have another type of assay in order to nail down which one is which. And so this is where the triplet binding assay came in, developed by Nuremberg and Leder, to determine other specific codon assignments. So ribosomes bind to single codon of 3 nucleotides, and then complementary amino acid charged tRNA can bind. So you have a ribosome, so this is what produces the polypeptide chain, and these can incorporate the tRNAs into the ribosome. And so if you have a synthetic RNA triplet that you were able to produce, so they produced a UUU, for example, then they were able to incorporate the ribosome together. So this ribosome structure was able to come together with the tRNA in one part and then the triplet codon in the other. And so they could use that in order to further identify codons that they could synthesize these small mRNAs for and associate them directly with the amino acids. So you can see they were able to fill in a large number of the trinucleotides kind of in this way. And so one of the things you should start to see from these experiments that the researchers certainly noticed is that there seems to be a lot of flexibility in terms of the last codon is most often to be the one that can be degenerate. And so what do we mean by that? So lysine, there's two different ways to make lysine, and if we look at the difference, in both cases it starts out with the same two, but this third codon can be different. Proline, what happens here? CC, 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 they all start with CC, but there's flexibility in what the last amino acid is. So this is not a rigid rule, obviously. So in some cases there is degeneracy just for two, so there's two ways to make lysine, and so AAC and AAU. Sorry, and this is best able to be seen in here. So we see, a lot of times there's this flexibility. It's not always true. So if we look at histidine, there is degeneracy in the third, but it's not a simple rule where it's CA and then all four can produce the same amino acid. So just remember that this is not like a rigid rule that says it doesn't matter at all what the third codon is, but there is a lot of flexibility there in either having two different ways to make a amino acid or having four ways to make an amino acid. So finally, researchers were also able to chemically synthesize long RNA. So these are short repeating sequences that are enzymatically joined, which made long RNAs. So they were able to produce, for example, a UGU together, or they could produce a GUG. And so they're able to produce this repeating triplet of UG, 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 UG. And so you would have half of the repeating sequence in this case was a dinucleotide UUG. So they could join the UGs together. And so by joining all of these UGs together, you could only produce two different types of triplets. So UGU or GUG. Similarly, if you used trinucleotides UUG and you just put them all together, one of the things you could do is modify the initiation sequence. And so if it initiated here, then you would get all UUGs. If it initiated here, you would get UGUs, and then here you would get GUUs. They also could form tetranucleotides, and then by combining the tetranucleotides together, again, they could produce another set of different triplets. And so they were able to, again, perform these experiments and further refine what exactly was produced by each triplet. So in the end, all these different experiments, lots of people thinking about this. So pretty quickly after the discovery of DNA, researchers were able to actually deduce the genetic code. And so now that has allowed us to take that DNA sequence and now predict a protein sequence for a large number of genes. So again, the genetic code is degenerate. Many amino acids are specified by more than one codon. Only tryptophan and methionine are encoded by a single codon. Genetic code shows order, so chemically similar amino acids share one or two middle bases in the triplets encoding them. And then importantly, this led to the wobble hypothesis. So the wobble hypothesis just says the initial two ribonucleotides of the triplet codes are often more critical than the third. And this is just where the degeneracy is. So because all of these triplets are able to produce the same amino acid, this is the wobble. So it just means that this last one doesn't really matter. So all that what matters a lot more are the first two. The initiation, they were able to determine that an AUG is an initiator codon. Initial amino acids incorporated into all proteins. So in bacteria, there's so the first amino acid is always a methionine, and in bacteria, there's actually a modified form of methionine. So it mostly looks the same, but it has an additional formal group on it, and this is referred to as FMET. Excuse me. So if the AUG, so if you have a long mRNA, the AUG at the beginning, this is almost always used as the initiator. This would incorporate FMET. But if you also had an AUG in frame later on in the mRNA, this would just be MET. So we kind of discussed this before, but AUG can be used either as an initiator or as just encoding of methionine. And so the contextual clues are kind of up here that determine, what exactly is done. But we'll get into that farther. And then the termination codons, UAG, UAA, UGA. So these do not encode for any amino acid. They're not recognized by tRNA. And then the translation terminates when these codons are encountered. So we also talked about the universality, whatever that word is, of the genetic code. And so this just means that if we look at all sorts of different organisms, so if we solve the genetic code in bacteria, if we solve the genetic code in humans, in general, we always find the same code. There is a small number of exceptions. So one of the really important exceptions is mitochondria. So mitochondria, we haven't talked too much about this in this class. Maybe you've talked about this in other classes. You should certainly know that mitochondria are the energy source, the fuel source for the cell. This is where ATP is created. They were thought to be, the mitochondria, the evolutionary history of mitochondria is thought to be a symbiosis event between two bacteria, where a smaller bacteria was incorporated by a larger bacteria, and that smaller bacteria eventually evolved to become the mitochondrial organelle. They have some very important differences. So these are codons. So in normal genetic code, AGA and AGG would encode an arginine. So in the mitochondria, these are also used for termination sequences. And then there are certain other, besides mitochondria, there's also some other organisms, mycoplasma, paramecium, where it's been shown that there's been some modifications of the genetic code as well. So where might you encounter this? So if you were studying a gene, so the mitochondria also has its own genome. It's also able to produce its own proteins. So you might encounter this if you're studying a mitochondrial gene and you look at the mRNA sequence, and you might be surprised to find this seems to make no sense in terms of the protein that is produced. And that's because you're probably using a normal genetic code, and you have to remember that you have to use the special genetic code for mitochondria if you want to accurately translate the mitochondria genome into protein sequence. So that is the extent of what we're going to cover on the genetic code. Hopefully this is all pretty straightforward. I think you've all seen it before, so hopefully this all made a lot of sense.